Welcome to today's lecture on the topic of fatty acid metabolism. In this session, we will explore the intricate details of how fatty acids are processed within our cells to generate energy. We will begin by discussing the structure and nomenclature of fatty acids, highlighting their importance in our metabolism. Fatty acids are long-chain hydrocarbon molecules with a carboxyl group at one end. They can vary in length and saturation, influencing their chemical properties. Fatty acids are typically named based on the number of carbons in the chain and the position of any double bonds. Before diving into the specifics of fatty acid metabolism, it is essential to understand the basic principles of metabolism and energy production. Our body constantly undergoes biochemical reactions to extract energy from the nutrients we consume, including carbohydrates, proteins, and lipids. Fatty acids play a crucial role in energy storage. When we consume an excess of calories, these excess nutrients are converted into triglycerides, which are composed of three fatty acid molecules attached to a glycerol backbone. These triglycerides are then stored in adipose tissue for later use as an energy source. Beta oxidation, the process by which fatty acids are broken down to generate energy, takes place in different cellular compartments, primarily the mitochondria and peroxisomes. The mitochondria are the major site of beta oxidation, while peroxisomes also contribute to the breakdown of specific types of fatty acids. To enter beta oxidation, fatty acids need to be activated by the enzyme fatty acyl-CoA synthetase. This enzyme catalyzes the ATP-dependent formation of fatty acyl-CoA, a high-energy molecule that serves as a substrate for subsequent steps in beta oxidation. Activated fatty acids, in the form of fatty acyl-CoA, need to be transported into the mitochondria, where beta oxidation occurs. This process involves a carnitine-dependent shuttle system that facilitates the movement of fatty acyl-CoA across the mitochondrial membranes. Beta oxidation involves a series of enzymatic reactions, with each step catalyzed by specific enzymes. Key enzymes in this pathway include acyl-CoA dehydrogenase, anyl-CoA hydratase, and several others. These enzymes play vital roles in the oxidation and hydration of fatty acyl-CoA molecules, breaking them down into acetyl-CoA units. Let's now delve into the step-by-step breakdown of fatty acid molecules during beta oxidation. Each cycle of beta oxidation involves the sequential removal of 2 carbon units from the fatty acyl-CoA chain, resulting in the formation of acetyl-CoA. This process continues until the entire fatty acid is completely oxidized. As the fatty acids are broken down, the energy released is captured in the form of ATP and reducing equivalents, such as NAD and FOD2. These energy-rich molecules then participate in subsequent metabolic reactions, ultimately providing the fuel necessary for cellular activities. The process of beta oxidation is tightly regulated to ensure energy homeostasis in the cell. Hormones, such as glucagon and insulin, play key roles in coordinating the rate of fatty acid breakdown. Additionally, allosteric regulators and post-translational modifications modulate the activity of specific enzymes involved in beta oxidation. Although both mitochondria and peroxisomes contribute to beta oxidation, there are distinct differences between their respective pathways. The length and saturation of fatty acids, as well as the fate of the generated acetyl-CoA, dictate the specific organelle responsible for their breakdown. Disruptions in beta oxidation can lead to fatty acid oxidation disorders. These inherited metabolic conditions result from a deficiency in specific enzymes or transporters involved in this pathway and can manifest with severe symptoms, including hypoglycemia, muscle weakness, and neurologic abnormalities. In conclusion, fatty acid metabolism is a complex and highly regulated process that serves as a vital energy source for our cells. Understanding the various steps involved in beta oxidation and the factors influencing its regulation provides insight into both normal physiology and pathological conditions. Embracing this knowledge allows us to better comprehend the underlying mechanisms and develop effective strategies for intervention when metabolic disorders arise.